welcome to the Current Science and Technology podcast from the Museum of Science in Boston. I'm your host, Corrine Tate, and every week we bring you interviews with guest researchers and our museum staff covering science and technology in depth. Advances in microscopy allow us to see some of the smallest structures inside cells. But biologists want to go further. They want to see the details of how the molecular machinery of living cells carry out their critical functions. The problem is that these tiny molecules are smaller than a wavelength of visible light, putting them beyond the reach of conventional light microscopes. Here to tell us how researchers are tackling this problem is Peter Kinnair, a microscopist and engineering professor at the University of Georgia. Welcome, Dr. Kinnair.
Hi, thank you. Thanks for the invitation.
My pleasure. I'm excited to talk about this. To begin, can you tell us a little bit about the limitations of light microscopes and how microscopists like you are getting past these barriers?
Sure. I guess to begin with, let me just say that the biologists use a type of light microscopy called a fluorescence microscopy. And the way fluorescence microscopy works is that you add a little fluorescent beacon to the molecules inside of your biological sample that you're interested in. And that allows you to know exactly what you're looking at in your sample. So you can tag different proteins and different structures inside the cells with different color beacons. And that allows you to really understand in detail, what you're seeing and what's going on. And that already was a huge advance for biological microscopy because if you just, for example, put a biological sample under a normal white light microscope, one that just shines light onto the sample, you're not going to really be able to tell what's what. So all the different things inside the cell look very similar optically, and you can't distinguish, say, one protein that has one function from another structure that might have some other function. So fluorescence microscopy was a huge advance for biologists to understand what they were looking at.
So in this case, you're basically lighting the cell from within, and you're lighting up the different structures with different colors so you can tell the difference.
Exactly.
I see. So what are the limitations of this?
The big limitation with microscopy is that the resolution is about 250 to 300 nanometers. And what that means is that in a normal fluorescence microscope, if you have two structures or two, say, small objects that are within 250 nanometers of each other, it's very hard to tell them apart. They're going to each produce a spot of light that's about 250 nanometers in diameter. And since those spots will overlap almost entirely, you won't really know that there's two distinct objects there instead of just one. And as you know, a lot of objects in a cell are very small and very close together. So this resolution is a big limitation for understanding things that are happening inside biological samples.
So microscopists, though, have found a way around that limitation. Is that right?
Yes. So over the past several years, there have been a few different techniques that have emerged that allow you to cheat in some sense and get resolutions much better than the resolution of a conventional microscope. So one of these techniques is called STORM, and that's the technique we've been using in the QSTORM project. The way STORM works is that, as I explained before, if you have two objects that are very close to each other, you can't tell them apart. But if you know you're only imaging one object, so if you have sort of one fluorescent molecule that's emitting light and that's the only thing that you're imaging, it will emit a spot of light that's 250 nanometers in diameter, but it's much easier for you to figure out exactly where the center of that spot is. So the spot may be 250 nanometers in diameter, but you can say much more accurately say that the center of the spot is then a 20 nanometer accuracy.
And the center of the spot would be the source of the light that you're using to tag an object.
Exactly. That's where the molecule is emitting from.
So STORM has a way, as you described, of pinpointing the center of each spot of light that you're imaging. Now, how does that, in the end, give us better resolution?
So then there's another technique or trick that you use then that combining with the fact that you can figure out where the center is allows you to get very high resolution. What you want to do is if you're going to label some structure inside the cell that you're interested in with many fluorophores, many molecules that emit light. And again, if you image all those molecules at once, you'll have a conventional image with a resolution that's not very good. But if you can image each molecule sort of sequentially And it doesn't matter what the order is, any random order. But if you can image them one at a time, then each time you image one, you figure out exactly where the center is of the emission, and you record that in a table, and then you go on and you image the next one. And once you've imaged all the molecules, you have this long list of where the center is for each spot of light. And then you plot the locations of those centers on a new image, and you get a much higher resolution picture of where all the molecules are. And that's what gives you the high resolution storm image.
Oh, wow. So you've just described this technique that can give us these pretty detailed images. What are some of the biological problems that researchers would need this technique for?
So I'm not a biologist, I should just state that, but I think there's a lot of different problems that people are interested in which this kind of resolution is important. One classic example that people have used a lot is that there's a lot of vesicles inside cells that are used to transport things in and out of the cell, sort of maybe to get rid of waste or to take in things from the outside that the cell needs. And these vesicles can be on the order of maybe 100 nanometers in diameter. So if you want to understand how these vesicles, what their structure is like, how they're built and how they encapsulate these different molecules that they take in and remove from the cell, you need resolution much better than a conventional microscope. So with a conventional microscope, one of these vesicles would just look like a spot. But with these new techniques, now you can start to see some of the structure. You can actually just distinguish the surface of these vesicles from what's inside.
So this technique can really give us a lot of insight into the real fine detail about how a cell works, how it's moving these cargoes around the cell, for example, in vesicles, or the actual structure of a living cell and how that changes.
Exactly. So you can really start to look at some of the structural detail of the cell now with enough resolution to really see it, as it is. And of course, people could do that before with electron microscopes, but electron microscopes are very hard to use. And one of the big drawbacks of an electron microscope is your sample has to be in a vacuum so that you can't look at living cells. You can't look at things change and grow in an electron microscope, whereas you can do that with a light microscope.
Oh, so this is kind of our window into dynamic living cells.
Yes.
And that's something that we've never had before. That's really exciting. So one question I still have about how this technique works is you mentioned needing to image each one of these light spots sequentially. So I understand that we're kind of lighting the cell from within with kind of glowing molecules, but how do we control them? How do we turn them on and off at different times?
Yeah, so that's a really important aspect of this imaging technique. Most fluorescent molecules, if you shine light on them, they will then emit light. And so they'll all emit light at once. So you need some mechanism for being able to make sure that only a very small number of them are emitting at any given time. So people have discovered specific fluorescent molecules where you can make them not emit light. So you're going to basically turn them off like a light switch and have only a few of them turned on. And that was a really important advance for this kind of imaging. So you can't make them turn off and on precisely. You can't say, I want that one on and all these other ones off. But you can basically say, I'm going to turn almost all of them off so that only a few are on. And then you can look at those few. And then you can again turn them all off again and make sure that only a few other ones are on. And so if you do this over and over again, you will build up an image where you've basically looked at all of the molecules over time.
But I understand that you're actually using an entirely different light source. You're using something called quantum dot nanoparticles with your storm imaging. Can you tell me a little bit about what these are and why we would want to use them?
We're collaborating with a chemist, Jessica Winter at Ohio State University, who's an expert in creating these quantum dot nanoparticles. So a quantum dot is essentially a very small piece of semiconductor. So semiconductors are materials that we use a lot to create light. And the material that emits light in a LED bulb it's called a light emitting diode, and it's made up of a semiconductor material. And so what a quantum dot is, it takes the same material that emits light in these new light bulbs, and it's a very small, maybe only like a 10 to 15 nanometer in diameter, well-organized crystalline sort of sphere. And one of the remarkable properties of these quantum dots is because they're so small, and you're basically controlling the wavelength of the light and the electrons inside the material, the size of the quantum dot will determine the wavelength of light that's emitted.
That's pretty cool. You're saying that the same material that we use in light bulbs, we can use those basically as little tiny mini light bulbs inside cells?
Yes, that's correct.
Why are these better than the other light sources that we've used with STORM?
Well, they have their advantages and disadvantages, but one of the problems, as I mentioned before, with the other types of molecules is they don't emit that many photons. So typically what will happen with, say, these fluorescent proteins is that they'll emit maybe 500 to 1,000 photons, and then they'll just sort of stop working. Something will change inside the molecule, and it'll just stop emitting light. So you want more photons to be emitted from the molecule. The more photons you get, the brighter the spot you have, and the brighter the spot you have, the easier it is to tell where the center is. So that really directly affects your resolution in STORM imaging. So quantum dots don't have the same problem of just spontaneously stopping to work, and they can potentially emit many, many more photons than a fluorescent protein or a small fluorescent molecule.
So how is it that you started working with Jessica? Where did this idea to use quantum dots with STORM imaging come from?
Well, both Jessica and I were invited to a workshop that was hosted by the National Science Foundation. And the purpose of this workshop was to get researchers who were interested in bioimaging together, and there was researchers from all sort of different areas of science. So there were chemists there, and there were computer scientists, and there were biologists, and there were instrument developers like myself there as well. The point was to get us all together and talk with each other to think about sort of interesting things that could be done and what the current problems are in the field and things like that. And Jessica and I were sitting talking at lunch one day, and I asked her whether she could make the quantum dots that she developed sort of turn off and on so that they would be useful for STORM imaging. So I had been aware of STORM imaging for some time, since it was a sort of very exciting development in my field of research. And I had been thinking about what kind of interesting things could be done. And so having the opportunity to talk with Jessica, I wanted to know whether she could make these molecules that we could then use for STORM imaging.
That's very cool that this idea for kind of transforming this technique into something even more powerful really came about by, sitting with Jessica and talking over lunch and kind of coming up with this new ideas about how to control quantum dots.
Yeah, it was nice. I think it's great to have those opportunities just to talk with people casually. It really where a lot of exciting things come from.
Now, I understand that you've actually gotten some inspiration from astronomy that you're going to use in microscopy. They sound like really different fields, so I'm curious how one can help the other.
They are very different fields, but they both use light to image things. And one of the problems that both astronomy and microscopy have is that the light that you want to image has to travel before it gets to the camera where you're going to create your image. So in astronomy, you have light that travels many, many light years. And by the time it gets to the Earth, the star is so far away that it essentially looks like a tiny point. And it should create sort of a perfect point on your camera. But as the light travels through the atmosphere, the tiny temperature fluctuations and all the atmospheric turbulence distorts that light wave. And by the time the light wave gets to your telescope, it gets distorted. So the image that gets created on your camera, it turns out to be sort of a much sort of bigger sort of blob of light than you should have gotten if everything had been perfect. In microscopy, what happens is you have some sort of fluorescent object. You've sort of tagged something inside of a sample that you want to look at. Say you want to look at nerve cells within a fruit fly. You add a fluorescent molecule to something inside the nerve cell, and then the light from that fluorescent molecule has to travel through the body of the fly before it gets to your microscope and gets imaged on your camera. And then the body of the fly has all sorts of different structures in it, which have slightly different optical properties. And that leads to the same problem as in astronomy. The light gets sort of distorted, and instead of getting a nice sharp spot from your fluorescent molecule, you get a fuzzy blob.
So how are you getting around that?
There's a technology that we're borrowing from astronomy. Astronomers have been working on a technology called adaptive optics for maybe since I think around the 1970s. What adaptive optics is it's a technology where you measure the wavefronts of the light. So light is a wave, so it has sort of peaks and valleys, just like a wave hitting a beach. And when those waves are going through your telescope, you want the waves to be flat. So you would like the waves to be, say, perfectly straight and parallel to the beach. But they're not. They get all curved and bent. So in adaptive optics, you measure how bent those wavefronts are, and then you use a special mirror where you can control the shape of the mirror to straighten out the wavefronts. So essentially what is happening is that some parts of the wave will be ahead and some parts of the wave will be behind. And with the mirror, you make the parts of the wave that are ahead travel a little further so that the other parts of the wave can catch up. And the parts of the wave that are behind, you make them travel a slightly shorter distance so that's easy for them to catch up. So then by the time that the light reaches your camera, it's got a nice flat, straight wavefront.
So your mirror is basically correcting the differences that are making the waves kind of distorted and straightening everything back out again.
Yeah, exactly.
That's pretty clever.
It is. It's very cool. So now we can take this adaptive optics technology that straightens out the wavefronts and essentially take exactly the same or almost the same designs that we use in a telescope and put them into a microscope. So then we can develop sort of much better images of things that are sort of inside of biological samples.
So it sounds like you've been bringing together a lot of different ideas to create a new imaging technique that's potentially a lot more powerful than what we have before. Could you kind of bring everything together for our listeners?
Sure. I met Jessica at this conference. And so along with two biologists, we got some funding to develop some of the ideas we've been talking about into a new type of storm imaging that would allow us to get higher resolution and deal with some of the optical imaging problems that happen when you get deeper into biological samples. The project that we have is called QSTORM, and the Q is for quantum dots, and the storm is for this storm imaging technique that gives you very high resolution. And so we're going to combine using quantum dots, and Jessica's developing quantum dots that we can switch on and off. And I'm developing a storm microscope that's going to have adaptive optics. And so by combining these two ideas, we hope to be able to take storm imaging and allow it to get sort of very high resolution images in sort of thicker biological samples where we have to deal with these sort of issues of the light getting distorted that I talked about. And we're going to use the quantum dots to make sure that we still have very bright molecular beacons inside of these samples. And then we have two biologists in the collaboration that are going to have interesting biological problems that want to study that we hope will be able to help them out by using QSTORM.
Well, thank you, Dr. Kinnair, for sharing with us this exciting new technique for biological imaging and shedding some light on how we're able to see more detail than ever before.
My pleasure. Thanks for having me.
If you'd like to hear more about Dr. Kinnair's work or the NSF-funded QSTORM research collaboration, please visit the team's website, qstorm.org.
That's it for this week's show, but be sure to come back next time for more of the latest in science and technology. This podcast is a production of Current Science and Technology at the Museum of Science in Boston, part of the Boston community for over 175 years. For more information, visit our website at www.mos.org/podcasts or email us at podcast@mos.org. Thanks for listening.